Hello everyone, welcome back to another lecture on regeneration biology. So today we will learn about zebrafish heart regeneration in a brief. And zebrafish, as we discussed in the previous class, has enormous regenerative capability. But every organs are not equal in regeneration. Like we know, liver can be regenerated in every animal. But when it comes to central nervous system, say brain and spinal cord, the regeneration ability is compromised in a lot of species. But when it comes to heart, because it's a very unique tissue, it is made of cardiomyocytes or the cardiac muscles, which is different from that of skeletal muscle. So every animal, when they undergoes the so-called reduced supply of blood to the heart muscle, that we usually call it as myocardial infarction, that damaged tissue responds by scar formation, which can debilitate the contractile ability of the ventricle. If auricles are damaged, the heart will continue to function, but if the ventricular muscles are damaged, the contraction is maximal and more powerful when it comes to the ventricles, and if that is damaged, the animal will not be able to supply blood to the rest of the body, and that animal may succumb to death. So animal models to study cardiac regeneration has been one of the interesting aspects or interesting topic in regenerative medicine, because everybody want to understand why and how the myocardial infarction takes the life of an organism. So lot of research is going on in understanding, can we avoid the formation of scar or the fibrosis after a myocardial infarction. So animal models to study the cardiac regeneration has been explored for various different questions. Permanent fibrosis and chronic deterioration of the heart function in patients after myocardial infarction, in short form it is referred to as MI, present a major healthcare burden worldwide. And in contrast to restricted potential for cellular and functional regeneration of adult mammalian heart, a robust capacity of cardiac regeneration is seen during the neonatal period in mammals. Neonatal means soon after birth, very early stage of the organism's lifespan, that is neonatal, and in the adults of many fish and amphibian species. Neonatal mammals and adult fish and adult amphibians are able to cope up with a damaged heart or an injured heart. But in mammals, when it comes to adult, the damage is not resulting in a regenerative response. So that is the time when you get some cardiac trouble, not during your neonatal phase. Hence, the knowledge or understanding or ability to intervene to cause a proper regenerative response in adult is very important when a mammalian species is concerned. However, we lack a complete understanding of why cardiac regeneration occurs more efficiently in some species than the other. So this is something which baffles the researchers. Adult phase of some animals, heart can regenerate after a damage, whereas adult phase of some animals respond to cardiac injury by scar formation. Why so? This is a baffling question to the researchers. The capacity of heart to regenerate after injury is controlled by a complex network of cellular and molecular mechanisms that form a regulatory landscape, either permitting or restricting regeneration. So in animals that can regenerate, the gene expression landscape is allowing the regeneration. The gene expression events are allowing the regeneration, whereas in the same tissue in another species, adult stage, the same landscape causes set of gene expression that do not allow, that means we call it as a non-permissive environment is created, hence regeneration do not happen. So this is a dendrogram you can see where mammals are grouped here, amphibians are grouped here, fishes are grouped here. And you can see the gray color box indicates low regenerative ability, red color indicates high regenerative capability, and it has been classified into three: fetal, neonatal and adult. Fetal is shown as triangle, neonatal is shown as circle, and adult is shown as square. So if you have a blue square means low regenerative ability, red square means high regenerative ability. So in mammals, you can see fetal is study done in sheep. That doesn't mean that others fetal is missing, means that it is not been explored. And when it comes to neonatal, you can see in rat, neonatal heart regeneration is excellent because red in color, and mus musculus it is high, and rabbit it is high, and human it is high. And human, of course, nobody injure human and test it. Okay, so you should understand any damage or any surgery that has to be done, it heals without any scar formation. That is what we know. And in also in pig neonatal stage excellent. Whereas in all these species which has got excellent regenerative capability, in adult phase which you see a blue mark, that means it do not have low regenerative ability, it do not have excellent regenerative ability, rather it has got a very minimal or nil regenerative ability when it comes to the adult. One exception is there, spiny mouse. What you can see here, it's neonatal is not been tried, rather you don't have to try neonatal spiny mouse because adult heart has got excellent regenerative ability. So it is logical to assume that neonatal and the fetal will have much more or much efficient regenerative ability. Unlike all the mammals tested, spiny mouse, adult heart has the ability to regenerate. When it comes to amphibians, nobody bothered to study about fetal and neonatal because adults have the excellent regenerative ability. You take newt, any newt of choice, you take it, and xenopus, that's African cloud frog, adult have got minimal regenerative ability. And whereas xenopus tropicalis, xenopus levis have got less ability, xenopus tropicalis have got adult phase also regenerative ability is there, and axolotl have got excellent regenerative ability of the adult. And when it comes to zebrafish, nobody bothers to study fetal and neonatal heart because adult regenerates excellent. The exception being Mexican tetra. It has got two forms, surface dwelling form and cave dwelling form. Surface dwelling form, adult have got good regenerative ability, whereas the cave dwelling form have got least regenerative ability. Remember, both comes under same genera, that is Astyanax mexicanus, that is the Mexican tetra. They are same species and same genera and same species, but surface dwelling form has excellent heart regeneration ability. Cave dwelling form, which is basically blind, do not have the ability to regenerate. It is something worth knowing. And Japanese Medaka do not have the regenerative ability at all, hence it doesn't have the heart regeneration ability also. Whereas the killifish have got excellent regenerative ability, and zebrafish, as you can see here, has got excellent regenerative ability. And many species, there is another species called the giant danio species that also have the regenerative ability. Goldfish has got excellent regenerative ability. So only the tetrafish, which is the surface dwelling form, can regenerate. Cave dwelling form it cannot, and Medaka cannot regenerate. So this picture lets you know that regeneration is not a blessing, but it can often give trouble in some organisms' survival. If it has the ability to regenerate, then it will make a better living for a further going forward. Whereas in mammals, you may wonder, it doesn't have the regenerative ability, then how the species is existing. So we cannot connect the regenerative ability of the heart to the survival of a species. Like you have seen some fishes which cannot regenerate. So it has to be understood that whether or not a species is regenerating, the ability to regenerate gives an added advantage to that particular species. So heart regeneration, when you look into, there is no significant regeneration of adult mammalian cardiac muscle after experimental injury paradigms. This efficiency is highly relevant to human disease that we refer to as myocardial infarction. And the scarring is the primary cause of morbidity and mortality. Like I told you, some time ago that the formation of the scar renders the cardiac muscle less contractile. So this is not a friendly thing to happen. So you want the heart muscle to contract, but the scar will come in between. It is just like you are eating some food and in the middle I am putting a stone and you are biting through the stone. How horrible feeling that will be. Same logic applies. When the heart is contracting, the scar will be a impeding, intervening obstacle. So there are currently several injury models that stimulate heart regeneration in zebrafish, including surgical resection of the ventricular apex, because zebrafish has got two-chambered heart and the ventricle has got a conical edge. It's almost like a inverted pyramid. So that cone is very easy to cut. You can either touch, handle everything without affecting the normal functioning of the heart. You can amputate it, or you can put a dry ice, and which is similar to that of the myocardial infarction, and that is called cryo injury. And people damage using genetic ablation also. Like you can express some gene called nitroreductase, which is from the bacteria, and then you can put a metronidazole, and the nitroreductase act on the metronidazole and kill the cell. So this is a genetic ablation. There are different methods are there. People use thymidine kinase ganciclovir system also to damage the cells. We don't have to go into the details, but most prominently used one is either surgical amputation or putting cryo injury, dry ice. So because dry ice mediated is something similar to that of myocardial infarction. Because myocardial infarction, nobody comes and cuts your heart, but rather the blood supply will be prevented. Whereas cryo injury mimics aspects of MI, that is myocardial infarction, genetic ablation produces massive injuries, that is removing up to 60% or more of the cardiomyocytes. That is why it is a very alarming situation, and it is not very logical or close to that of myocardial infarction that occurs in human. So one could use those genetic ablation studies for academic purpose, but may not have a clinical relevance. Inducing signs of end stage heart failure. If 60% of the cardiomyocytes are damaged, it is indicating of the terminal situation of heart damage. Unlike severe heart failure in humans, these signs regress within weeks, and the animals typically make a full recovery concomitant with muscle regeneration, where in those animals we saw in the previous chart, which is capable of regenerating the heart, they will completely restore. Doesn't happen in human. Heart regeneration involves two fundamental components. What is the component one? That is the proliferation of existing cardiomyocytes. Heart is made of cardiomyocytes and the cardiac muscles. That is the cells of cardiac muscles are called cardiomyocytes, and as the primary cellular source, and they are proliferatable and they can proliferate to some extent. And then that is not the only thing. An environment that stimulates the muscle regeneration from this source. Cardiomyocytes are proliferating, but are you able to make muscles, the contractile muscles? That is what is important. You have raw material, but are we making the end product? So both are needed. Raw material has to be formed. Cardiomyocytes, if they are not able to proliferate adequately, then they undergo reprogramming, and they become stem cells, and they proliferate in number, and then they differentiate into cardiomyocytes and cardiac muscles. So heart regeneration is, people have done some thing called genetic fate mapping experiments. Genetic fate mapping, which basically means, if I give a simple example, you will understand. You lit an agarbatti, or you put a smoke, you created a smoke in your house, and you can see where all the smoke is going. But normal air also moving like that. But you can't see, normal air is not colored. So what you did, you put a tracer in the air in the form of smoke. So if you lit a agarbatti that gives a smoke, and you can see where all the smoke is going until certain distance. You cannot you put an agarbatti on Monday and you cannot trace that smoke on Wednesday. That is not possible because it diffuses out. The law of diffusion applies. Same way, if you have a cell, origin of cell, and you mark them with something, there is today if this cell proliferated, I am marking it with a GFP or some protein which is traceable. So it will become a permanent marker. Like you may have seen in some people who go to some temple like Tirupati, or maybe going to a large pilgrim spot, the law authority will give you some sticker on your hand. So that will tell you that, there will have a barcode or when you entered and how long this is valid, etc. So wherever you go, you have a tag. That is, you are a bona fide, you are an authenticated visitor. And anyone can scan it and know, oh, this fellow entered this time and he will leave this premise in this time. Same way, genetic fate mapping allows researchers to understand, okay, the cell is formed today and where the cell is going to go eventually. Just like how will you know a kindergarten child will become a doctor or a lawyer or an engineer? You have no way of knowing. But if you have some tag in that stage, and if he become engineer, you will know, oh, this tag as a kindergarten I put it and he is having this tag now also. He became an engineer. That means you are detecting it. That is what we will do. Fate mapping experiments in zebrafish have made it clear that the regenerative ability of the zebrafish heart relies mainly or exclusively on the proliferation of the existing cardiomyocytes. The heart has got cardiomyocytes. They have to enter the cell cycle. Normally they don't proliferate. They have to enter the cell cycle from the G0 to G1 of the cell cycle. It has to migrate. These source cardiomyocytes show characteristics of dedifferentiation, including a reduction in the contractile structure. Cardiomyocytes, which are basically the cells of the cardiac muscle, they have the ability to contract. But when they get into a dedifferentiation mode, they do not contract. Because if they keep contracting, they cannot proliferate effectively. So they reduce their contractile property and they have more of their proliferative property and increase in number, and then they become this contractile cardiac muscles. So cardiomyocytes proliferation occurs at a low rate in the adult zebrafish heart, no matter whether there is an injury or not, but is sharply increased after a injury is made, or it is because of an injury response or a tissue damage happens. This proliferative rate of the cardiomyocytes increases tremendously, and because of which you are able to create a mass of cells, something similar to that of the blastema, what we have been discussing in planaria, zebrafish fin, etc. So injury to the zebrafish heart initiates an organ-wide reaction detectable as induced expression of a gene called RALDH2. What is RALDH2? A retinoic acid synthesizing enzyme. Other word, it is called retinaldehyde dehydrogenase. So retinoic acid is what? Vitamin A. We saw in the fin regeneration also. So RALDH, when it is induced, it tells that there is abundant supply of vitamin A on that spot, because that is needed. You need to have retinoic acid. It is as early as 1 hour of post-injury. Within 1 hour of injury, you end up getting a severe induction or a huge induction of RALDH. Otherwise, you don't get it. So why RALDH? You need to have the retinoic acid production, where the endothelial lining of the lumen. There is endothelial lining, that is the wall of the heart that is facing the blood. Extreme, that is where actually it is bathing with the blood, that is the endothelial lining of the lumen of the heart. Within a day or two of the injury, the epicardium, the outer lining of the heart, shows an analogous organ-wide response of RALDH2. That means you may have made a cut in one area. Say if the heart has got say 100 regions, you cut only in the 100th region, but the induction of RALDH will be from the sector 1 to sector 100. So every area, irrespective of where you caused the injury, the entire heart now produce the RALDH production. This doesn't mean that the whole heart is regenerating, but the response of the heart is pan heart, or it is spanning the entire heart. Induction. Then the epicardial cells proliferate and surround the regenerating muscle. Epicardium is the outermost layer. Okay. Epicardium and myocardium and the endocardium is there. 3 layers are there. Next picture will have better images of that, where they release signals that facilitate cardiomyocytes proliferation. So we can see the epicardium is the one which is allowing the RALDH expression, and the epicardial cells get into a proliferative phase and surround the regenerating muscle, where they release signals that can facilitate the cardiomyocytes proliferation. You can see here in this picture heart regeneration. you can see this is an uninjured heart. This is basically the SA node, AV node, those you call the pacemaker region. And you can see there is soon after injury there will be blood, and blood will get 6 hour post amputation, 6 HPA. You have created a cut here in this picture, and the endocardial activation happens, and then later epicardial activation happens by 3 day post amputation, and because of which you have upregulation of retinoic acid because RALDH2 is activated, TGF beta, sonic hedgehog, platelet derived growth factor PDGF, insulin like growth factor IGF, fibroblast growth factor FGF, and hypoxia factor. These are upregulated, and some microRNA such as MIR133 is downregulated. By 7 dPa is the peak of proliferation in the amputation method, and by around 14 dPa there is vascularization of vascularization means the blood supply or the blood vessels are being formed, and the muscle regeneration that happens by around 14 dPa, and then comes the electrical coupling and also the dissolution of the clot by around 30 dPa. Electrical coupling means the pacemaker should be able to give signal to these newly formed cardiomyocytes so that they can contribute in contraction. So the blue color is the clot, as you can see here, and the brown color is the blood vessels that you can see here, and RALDH2 mRNA is this sky green color. So you can see here its expression. Initially you see 6 hours post amputation, the entire heart is having RALDH, that's why it is blue in color, and the activated GATA4, which is a cardiac transcription factor regulatory sequence, you can see in green in color, as you can see here. Here also it responds up to here, and then it is restricted to the site of injury by around 30 dPa. You see little bit of this GATA4 expression, which is a heart specific transcription factor. So this is the layers of heart what we have seen of the heart wall. This is the epicardium, and this is the myocardium, where the cardiomyocytes and the cardiac muscles are there, muscular middle layer which is very thick, and the endocardium, the thinner layer. That is what we said, the lumen of the heart, or the lining of the heart to the blood, that is the inside. So this is a small portion. This is not a zebrafish heart, this is a human heart, but it's a cartoon just for the understanding. Same layers are being restored and retained in zebrafish also. So epicardium is the one which is contributing to epicardial activation, which contributes to these signaling events. Initial activation is the endocardial activation. That is what you are seeing here in the very beginning. So heart regeneration, when you look closely, it is a very interesting aspect. What we can reveal is, if you tweak, if you prevent some signaling event, like you saw in this previous picture, there are so many are there. RA is there, TGF is there, SSH is there, PDGF is there, IGF is there, FGF is there. So when this many are there, we think, oh, if I can get rid of one, what may happen? Oh, that many are there, no? It is not like that. You have apple, orange, banana, and or maybe other guava, so many fruits are there. If I remove one fruit, you will not starve. But in this angle, they are not like a fruit or a vegetable basket. Each one has got a role. So they are not expressed for a fashion. Each one, if you get rid of RA, you will find something. If you get rid of IGF, you will find something. That means heart regeneration may happen, but it is defective. Say, think about a human baby that is being born. Everything is fine, but eyes are missing. So you won't call it is a human baby only, but it is not a perfect baby. That means living of that particular baby will be compromised. Say someone don't have ears. So you may wonder, oh, it is still human. Yes, it is human, but it is not perfect. So if you get rid of any of the signaling event, sometimes it may not regenerate at all. Depends upon what is the hierarchical position of that particular signaling event. Is it sitting in the base of a branch of signaling events? Is it a master gene versus it is a player gene? But understand, every gene has got a role. That is what we will be seeing by blocking some pathway. What do we get it? So you have done the amputation, and what you are seeing here, soon after there is a proliferation, there is a key given here. Undifferentiated progenitor cells are here, and the green color is the differentiating cardiomyocytes, because undifferentiated cells can differentiate back. And here you are able to see this newly differentiated cardiomyocytes. and differentiated and newly differentiated. Both are functionally same, but depends on the age of them. And new vasculature, which is shown in red color, this vessel, and cells derived from the epicardium is shown blue in color here. As you can see here, 1, 2, 3, 4, 5, 6 different stages. This 5 and 6 something important, because 5 and 6 is somewhat towards the terminal phase of regeneration. What they have done is they just got rid of FGF signaling. Remember, FGF signaling is listed here. You can see here FGF signaling is there. And they got rid of the FGF signaling, and what they end up getting? They end up getting a scar formed. Zebrafish that is capable of regenerating, just you got rid of the FGF signaling. By blocking the FGF signaling, wound healing happened and heart is fine also, but there is formation of a scar. That means contraction may not be perfect. Although regeneration happened, contraction may not be perfect. Why? You got an unwanted scar formed in between, which can interfere with the proper contraction of the cardiac muscle. So, cardiac muscle characteristics, if you look very closely, it is something very interesting. Cardiac muscle characteristics, they are associated with different types of heart regeneration depending upon which species you are talking about. What we have seen so far is many animals, based on their fetal, neonatal or adult, depending upon that there is a variation. Mammals, invariably majority of them, neonatal and fetal, have the ability to regenerate, not the adult. Exception is for the spiny mouse, which is of Acomis species. Whereas when it comes to fishes and frogs, nobody bothered to study regeneration ability of fetal or neonatal, but adult can regenerate. Exceptions are Medaka or maybe some cave dwelling form of fish, etc. Now, how are they responding at tissue level? Mus musculus, that is house mouse, versus spiny mouse. If you compare, increased neovascularization in higher numbers of cells and the fibrotic wound tissue and the elevated number of diploid mononucleated cardiomyocytes are associated with increased cardiac regeneration potential in the spiny mouse compared to that of house mouse. You can see here in this A panel, you can see this is mus musculus, this is Acomis species, and the ECM is very strongly formed, and the cardiomyocytes that are formed, and you have a huge energy consumption happens, and also there is formation of an unhealed area. That means there is a scar that is being formed. Whereas in the case of spiny mouse, you can see the cardiomyocytes that are formed and the fibroblasts formed are not contributing to the formation of the scar. Same thing when it comes to the Medaka or Japanese rice fish, Oraceus latipes versus zebrafish. Oraceus latipes cannot regenerate, zebrafish can regenerate. So interesting thing, you can see, you injure it, the Medaka, and you have got a lot of this neutrophils that is being formed, and the macrophages are being formed, and you have some healing but no complete regeneration. Whereas in zebrafish, you have the same neutrophil cells and also the macrophages much more in quantity. Here the inflammation is very high, here the inflammation is minimal in zebrafish, and the macrophage, which is anti-inflammatory, is very high, hence there is no scar, complete normal regeneration happen. And in the presence of the restricted presence of neutrophils and the early recruitment of macrophages is associated with high cardiac regeneration potential in zebrafish compared to that of Medaka. Now let us come to the cave dwelling fish. Very interesting. Cave dwelling versus surface dwelling forms of the Mexican tetra, Astinax mexicanus. Elevated energy metabolism, restricted immune response and reduced fibrosis are associated with high cardiac regeneration potential in the surface dwelling form, not the cave dwelling form, compared to the cave dwelling form. You can see here their energy metabolism, and also you can see their immune cells. In the cave dwelling form, the immune response is strong compared to that of the surface dwelling form, and because of the low immune response, you are able to get a proper regeneration. So we will study about another topic in the next class. Thank you.